So last time we covered the different segments of the digestive system, the alimentary canal, and then we will start now to talk about the digestive system accessory organs. So we have here the pancreas, which is part of the digestive system and part of the endocrine system. This gland has a shape like if it was a hammer, so this is the head, that it is embraced partly by the duodenum. So this is the duodenum, the first part of the small intestine. This is the body. The most part of the pancreas is made by the body, and then we have the tail. The tail is intraperitoneal while the rest is retroperitoneal. So it's located just behind the stomach. And then the pancreas, 99% of its tissue is exocrine. So in the exocrine portion, we have this acini. If you magnify a piece of the pancreas, you will see this kind of honeycomb structure that we call the acini. And in the walls of the acini, we have these cells that are simple cuboidal cells that secrete the pancreatic juices, which includes water, bicarbonate ions, and proenzymes. These proenzymes are called cymogens, are inactive enzymes that help to digest the nutrients that we ingest, like glucose, proteins, and then carbohydrates, proteins, and then lipids. So the pancreas has a long duct that is called the main pancreatic duct. And in this main pancreatic duct, you carry the enzymes into the second part of the duodenum, that basically this duct empties in this major duodenal papilla, or sphincter of Oddi. So this duct receives then the enzymes that are produced here, and then these enzymes travel from the lumen, this is the opening, the lumen of this acini, into these ductules. So these are small ducts that they join, and then they form this main pancreatic duct. Now, we have a secondary or an accessory duct that is smaller than the primary or the main. So this one emerges in the head and empties into this other papilla, the minor duodenal papilla. So there is a reason for why we have a secondary duct. Just in case that you have an obstruction of this primary duct, because maybe you have a gallstone traveling through this common bile duct and obstructing here, or maybe you have a tumor in this area that obstructs this duct, you can still have proenzymes from the pancreas released into the duodenum, and you can still have digestion. Now, what happens in some cases? Well, in some cases, these can become obstructed, both of them, and then you will have these enzymes activated within the pancreas, and you have autolysis. So you can have pancreatitis. So the pancreatic tissue can be autodigested. And of course, this is not good. Now, then 99% of the tissue of the pancreas is exocrine, while the other percentage, which is 1%, is endocrine and is made by these pancreatic islets or islets of Langerhans, in which we have alpha cells, beta cells, and delta cells that secrete different hormones. Alpha cells secrete glucagon, which increases blood glucose levels when someone is fasting. And then we have the beta cells that secretes insulin that decreases the blood glucose levels just when someone has ingestion of glucose or, well, a meal. And then we have somatostatin that it is produced by the delta cells that prevents over secretion of insulin, and oversecretion of glucagon, regulating these two hormones to maintain a balance of blood glucose levels. So if you magnify this region, you can see what are the secretions from these cells. So here we have the cells of the acini that secretes these enzymes, which are lipases, and they can be as well chymotrypsin, trypsin, which are inactivated, elastases that helps to break down proteins. So as a generic name of the enzymes that break down proteins are called proteases. So these proteases are released as proenzymes by the cells of the acini, and then you also release these bicarbonate ions. Now, everything happens under the influence of two hormones, secretin and cholecystokinin, which are released by the duodenal cells. So these two get activated when you have amino acids here in this, or are released when you have amino acids in the duodenum, when you had a meal rich in proteins, and as well when you have meal rich in lipids. So you're releasing this cholecystokinin or CCK and secretin. They go into the bloodstream, and then these cells of these acini start secreting the enzymes to break down these nutrients. Now for the liver is the largest gland that we have in the body, and is located in the right upper quadrant. Anteriorly, this is the anterior view. What you can see this major lobe, the largest lobe in our liver, which is the right lobe. Underneath the right lobe, we have a storage for bile. This is the gallbladder. And then we have in the border of this right lobe this ligament that is called the falciform ligament that holds the liver into the anterior abdominal wall internally. And then to the left side of this ligament, the falciform ligament, we have the left lobe, and then underneath this ligament, we have the round ligament. Now, in the posterior view, what you can see, posterior and inferior view, what you can see, it is, again, the left lobe, the right lobe, and then you can see minor lobes. So between this round ligament and the gallbladder, you can see a lobe that is small, that looks like a square, that is called the quadrate lobe. Quadrate, cuadro in Spanish means square. So this is the quadrate lobe. And then just next to this lobe, we have something that is called here porta hepatis. The porta hepatis is basically the hilum of the liver. That's where you have the exiting of the two bile ducts from the liver, the left and the right, that merges to form the common bile duct. And then here we have the cystic duct, which is the one that brings the bile from the liver into the gallbladder, which the gallbladder has like this shape that looks like a little inflated balloon with the neck here, the body, and the fundus. This is a muscular organ that contracts and relaxes to mix the bile so that the components of the bile don't precipitate and you don't form gallstones. Now, the other component of the porta hepatis is not only this common hepatic duct, but also this hepatic artery, which is branch of the celiac trunk. So this is the hepatic artery proper, and then we have the hepatic portal vein, which is the one that brings the blood from the intestines, the jejunum and the ileum, that it is rich in nutrients so that the nutrients can be processed here by the liver. And now anterior to this porta hepatis, we have a long lobe that it looks almost quadrangular. This is called the caudate lobe. Caudate means tail, and it looks like if it was a short tail from a dog. And then posterior, here in this region, we have the inferior vena cava that receives the blood from the hepatic vein. Internally, the liver has these segments that we call lobules, just like the lobules in our lungs that are subsegments of the lobes. And these lobules, they have a hexagonal shape with a central vein in the middle. And then in the vertices, you have a triad, the portal triad. In this portal triad, you have a branch of the hepatic artery. So this is the hepatic arteriole. You have a portal venule, and then you have a bile duct. And then in between the central vein and these portal triads, you have rows of cells of the liver. The cells of the liver are called the hepatocytes. And these hepatocytes are very active, basically metabolizing the nutrients, sometimes storing them, the nutrients, like when you eat excess of glucose, that glucose is going to be stored in the liver as glycogen. And let's say that you spend a few hours fasting, your blood glucose level starts dropping. As it starts dropping, this glycogen is going to be utilized to bring the blood glucose levels up through glycogenolysis and glycolysis, so you will have energy. So then the liver can temporarily store glycogen. And then the liver makes most of the proteins in our body, with the exception of the antibodies. And then it releases them into the sinusoids, which are located in between the hepatocytes. Now, in between the rows of these hepatocytes, you have these venules, or these sinusoids, that brings the blood from the portal triad into the central vein. And then these arterioles of the hepatic artery proper, it will also bring blood towards the central vein. And this blood, of course, is going to be oxygen-rich blood and nutrient-rich blood that is going to provide oxygen and nutrients to these hepatocytes. Now, the hepatocytes, they are going to start metabolizing again all of these nutrients, storing some of them, including vitamin A. Vitamin A is one of these vitamins that is stored in the liver. We also store iron in the liver. And as some of these pigments from the catabolism of the erythrocytes are formed, this bile, or this biliverdin, will be going into this bile canaliculi, and then this bile canaliculi will bring the bile from this area, from the central vein, towards the edge, like from in this direction, so opposite to the blood supply. And then the bile will go into these bile ducts. Your right and your left lobe are going to have these specific bile ducts, so you call it the right and the left, and then they merge and they form that common hepatic duct. And the common hepatic duct is going to merge to the cystic duct, which brings the bile for temporary storage into the gallbladder. And then when this cystic duct merge with the common hepatic duct, they would form the common bile duct. And then the common bile duct, together with the pancreatic duct, empties into the second portion of the duodenum, at the major duodenal papilla. And the function of the bile basically is going to be emulsify fats. I told you this previously. So it's like when you're doing dishes at home. If you're doing it by hand, basically that soap that you make, if your plate has a film of fat, of lipids, if you put some of the soap there, the lipids are going to separate. So that's emulsification. Why we emulsify the lipids? Well, because you need to process them. The enterocytes, the cells of the intestines, they cannot bring in large droplets of fat, so they need to break it down into smaller portions. And then that's why you have emulsification. After you emulsify, your pancreas will release the lipase, and the lipase will break it down into smaller components so that you can form these lipids again in the enterocytes and then release them into the bloodstream. So here is a retraction of the right lobe, so it can show you the gallbladder. So again, this is the neck, body, and fundus, and this is the cystic duct that merges to this common hepatic duct, which comes from the anastomosis of the left and the right hepatic duct. So forming the common bile duct that again joins this main pancreatic duct, and then you have your emptying of bile and pancreatic juices into the second portion of the duodenum at the hepatopancreatic or major duodenal papilla. And what it controls the release of this? The opening and closing of the hepatopancreatic sphincter or sphincter of Oddi. Questions? When someone has removal of this because the person has gallstones, well, the person cannot metabolize lipids as easily as before, but they can receive some pills that has lipases that helps them to metabolize lipids. But that's one of the first things of the diet that they remove from the person. If the person doesn't follow that diet, the lipids are going to appear in the stools. This is called steatorrhea. And then it would cause a little bit of uncomfortable sensation in the digestive system. So, what are gallstones? Are lumps of calcium salt and cholesterol. If large in one of the ducts, causes abdominal pain. Sometimes the gallbladder can also be infected by bacteria. Not necessarily gallstones causes pain, also cholecystitis. And in this case, the person will have also pain in the right upper quadrant. Typically, the treatment is surgery, surgical removal of the gallbladder. It is done mainly by laparoscopy nowadays, but if laparoscopy fails, it can be done open. And then that may temporarily, again, reduce the ability to digest fats. And this is next raised view of the gallstones. So the gallstones sometimes can be radiolucent and sometimes can be radiopaque. Radiopaque means that you have the denser tissue in your body looking whitish. So in this case, you can see all of these gallstones there. Okay, so for the secretion of bile. So you have your duodenal cells. The duodenal cells, when they detect the presence of lipids in the duodenum, they release cholecystokinin. Cholecystokinin activates this contraction of the muscle around this gallbladder or within the walls of the gallbladder, and then that contraction leads to the release of this bile. And then the bile will go and emulsify the fats, and then you will have the lipase acting on these fats, and then you can process. Another thing that it can happen is that you have the release of a hormone that is called secretin by your duodenal cells as well, and that along with the bile salts will be reabsorbed by the cells in the ileum, and that will lead to the release of more bile as you keep having some of these lipids inside this area. So it is like a positive feedback. So you start with very few bile release initially by CCK, but then secretin will be going into the bloodstream, activating your ileal cells, and then you'll have more bile release. So here is the summary of the accessory organs of the digestive system. So your teeth helps for mechanical breakdown of the food, so that's mechanical digestion. We also have salivary glands that helps in the chemical digestion. We have lipase enzyme and amylase enzyme in the oral cavity that helps to digest lipids and starch. And then the pancreas will do chemical digestion with its different enzymes like proteases and then amylase and lipase. Now in the stomach, which is not shown in here, we also have lipase. So we have three lipases: the salivary, we have the stomach lipase, and then we have also the pancreatic lipase. And then when it comes to amylase, will be salivary and pancreatic. And then we have emulsification by the gallbladder and, well, by the bile that it is stored by the gallbladder but produced by the hepatocytes. Now, something that I forgot to mention about the liver in the histology is that we have some cells that are called the Kupffer cells. So these cells are macrophages. Part of this is the function to destroy microorganisms, and also helps to destroy and recycle old or damaged red blood cells. The primary organ that does this is the spleen, but that doesn't mean that the liver cannot do it. So that's the Kupffer cells. When it comes to digestion, basically most of the secretions of the digestive system have a water component. And this water component, it is part of the gastric and pancreatic juices. So if you remember, hydrochloric acid formation has different steps. So you have, for instance, HCl, which is hydrochloric acid, formed when you have bicarbonate plus hydrogen ions merging to form ACO3, which is carbonic acid. So this carbonic acid will be converted into hydrogen ions plus bicarbonate. Let me put it this way so it doesn't get confused. So you have in your bloodstream bicarbonate, and then when you add a hydrogen, you will form this carbonic acid, and this carbonic acid can be converted into bicarbonate and hydrogen ions. So it's going in both directions, and this molecule of hydrogen will exit into the lumen of the stomach, and then you will combine it with chloride ions that diffuse through these parietal cells. So you join this with chloride in the lumen to create HCl. So if you recall, I told you about why we don't have the production of HCl inside the parietal cells. Do you remember why we don't do that? What will happen if you produce inside the parietal cells this hydrochloric acid? Will the cells be happy? No, you will destroy them. It's an acid. So then in the lumen, because you have this coat of mucus, you can bring now together the hydrogen ions with chloride. So, but then how you can start this process? Well, part of this is actually joining water with CO2. So that is what is going to give you also this. And then this enzyme that does this process, actually this one is ACO3 guys minus, this is called carbonic anhydrase. This is the same enzyme that you saw before in the respiratory system, and that you will see again in the urinary system. This enzyme is very important. So through this process, which is combining the CO2 from the bloodstream with water, you form this carbonic acid, and then this carbonic acid can be converted into bicarbonate, and then this bicarbonate can be broken down again into CO2 and hydrogen ions, so that the hydrogen ions can combine in the lumen of the stomach with this chloride. Now, in the case of digestion of proteins, so you have here two amino acids joined by a peptide bonding here. Since you have two amino acids, this is called a dipeptide. So one of the processes of breaking down proteins is by adding a molecule of water to break down this dipeptide link. So the OH, the hydroxyl group of water, will combine with this amino acid. In this case, this is alanine. And then the hydrogen ion is going to combine with this amino group from glycine. So this is called enzymatic hydrolysis, because you use water. Hydro means water. Lysis is breaking down. So this is a chemical digestion that happens to break down these dipeptide bonds. And then something that happens in these intestinal cells is that once you break down these molecules can move from the lumen of the intestines into the cells, passing through the plasma membrane in the apical site. So this is the apical site of the cell, this is basal site. And they don't cross through here because in here you have these tight junctions. So it's like a seal. It forces these molecules to go through here. And then you can have passive diffusion or facilitated diffusion. You can have channels in here or carriers. Example, for instance, we have sodium glucose transporters that co-transport glucose and sodium at the same time. And then sometimes some molecules, like in the case of lipids, will be reassembling here, and then they get transported into the lymphatics, into the lacteals. But sometimes it's just going directly without being metabolized into the bloodstream. And then whatever nutrients you have ingested, it would show in your bloodstream. Vitamins, carbohydrates, lipids, etc. So, we have example of carbohydrate digestion in here. You have a polysaccharide, like a long polymer with different molecules of carbohydrates. Can be glucose, can be lactose, can be sucrose. And then these ones will be broken down into oligosaccharides first, so into shorter chains. And then you can have, after you break down these oligosaccharides, you can have monomers like glucose or galactose, and you can have dimers like disaccharides. So then for glucose and galactose, they will be co-transported with sodium. And then fructose will be absorbed with facilitated diffusion. So this one, you don't need to have sodium available to absorb fructose. And then that is what is happening at the microvilli. But then here in the basal side, no matter if it's glucose or galactose or if it's fructose, everything goes in the basal side directly into the bloodstream by facilitated diffusion. While in here you have to have this co-transporter, and then in here you have a sodium-potassium pump that helps to bring sodium in so that you can use it with these co-transporters. If you remember, or well, if you have seen in some of these oral electrolytes that are sold commercially, you can see that they have glucose in it and then they have sodium. Is it contradictory? No. It is just trying to facilitate the bringing in of these electrolytes so you can bring them together when you just run out of that. And at the same time, of course, the glucose is going to give you energy to your cells. So that's the digestion of carbohydrates. Now for amino acids, you have a long polymer of amino acids. So this is polypeptide. This polypeptide is going to be broken down into into these oligopeptides. And then this process starts by the action of pepsin in the stomach. So we have pepsinogen that when it combines with hydrochloric acid, you have pepsin, and pepsin starts digesting your proteins into polypeptides, and then you have some enzymes from the pancreas, the brush border enzymes that are located as well here, that breaks down these oligopeptides into free amino acids. And then your free amino acids will be co-transported with sodium in the apical site. So this represents this microvilli. And then these amino acids will diffuse through the cells. This is the cytoplasm of the enterocytes. And then cross this basal part, the basal membrane, basement membrane of your enterocyte by facilitated diffusion, and then enters into the bloodstream. And then you have a sodium-potassium pump that helps to extrude excess of sodium into the interstitial fluid, and then potassium comes in. And now here we have the structural formula of bile salt. So it is made by this chemical structure with several rings that looks like cholesterol. So actually bile salts are cholesterol-based. And then it has this polar hydrophilic portion. This is non-polar. So this is the lipid part that do not interact with water, while this one is the part that interacts with water. So this is how bile salt looks like. So this will help to break down the lipids. And then it is not shown in here, but lipids starts being broken down in the oral cavity with this lingual lipase. And then we have in the stomach a lipase that it starts breaking down. It's called gastric lipase. These lipid globules into lipid droplets, like triglycerides can be broken down into diglycerides and free fatty acids. And then part of these globules can go into the intestines because we have a limited amount of lipase in our stomach, and some of them, they can be large. But we have the bile salts that comes from the bile that is released by the liver that can break down these globules into droplets. So it can break down these triglycerides into diglycerides and free fatty acids. And then through this emulsification, you form like these chylomicrons that have this central part made out of hydrophobic molecules. These will be the lipids, and then you have the bile salts that coat it, that it has this polar side that interacts with water, and they can be absorbed by the enterocytes, and the enterocytes will have this lipase that breaks down these chylomicrons into free fatty acids and monoglycerides. And then in the bloodstream, these will be absorbed. And then you have the formation of micelles, which will be like the monoglycerides and the free fatty acids surrounded by bile salts. And then for the lipid absorption, so you have your micelles that will be absorbing here, and then you will form them back again into lipids, and then you will coat them with proteins so that they can be transported in the lymph. So this is what we call the apoprotein coat of the chylomicron. So inside these chylomicrons, that's where you have your lipids, your triglycerides. And then this is going to be exocytosed and then enter into your lacteals. I told you that sometimes it is easy to see in the bloodstream when someone didn't have a fasting period when they take a blood test. So the plasma of someone who's not fasting, typically, if he has large amounts of lipids in the bloodstream, it will look like whitish instead of clear color. So that's how you can tell. So then these micelles will be absorbed, and then you can form these chylomicrons that will be entering into the lactose. So then what digests these carbohydrates? Well, will be your amylase enzyme that breaks down, in this case, starch molecules. Like if you ingest corn or potatoes, they are rich in starch, so these are the long polymers of glucose. They can be broken down into oligosaccharides, and then once this reaches the duodenum, the amylase from the pancreas will break it down into small oligosaccharides, and then in your intestinal cells you have brush border enzymes that breaks them down if they are disaccharides like lactose, which has galactose and glucose, into galactose and glucose, and then you absorb them. For the proteins, you start digesting them in the stomach with the pepsin. These proteins will be converted into these polypeptides. And then with the pancreatic enzymes, trypsin, chymotrypsin, elastases, you break them down into oligopeptides, and then you have your brush border enzymes like carboxypeptidases that breaks them down into individual amino acids. You absorb them, and then you make out of these amino acids new proteins. Your lipids will be emulsified, and then they will start to be digested with lingual lipase, gastric lipase into small globules, and then you emulsify them, you form micelles that will be absorbed, and then you will form chylomicrons that will go into your bloodstream. And then depending on if you need them or not, they can be used as a source of energy or can be stored for further use. So here is the table showing you the different enzymes and their actions. And again, when it comes to these pancreatic enzymes, which come from the pancreas in the pancreatic juice, which includes bicarbonate, water, and all these enzymes. So these enzymes will be inactive. This is what we call the zymogen. And this works with a neutral pH, not an acidic pH. So you have chymotrypsinogen and then trypsin. In the case of trypsin, this one will be activated by an enzyme that is called hexokinase. And then you start a cascade in which some of these will activate the other ones. And here are the products of the reaction. Dipeptidase and tripeptidase are found in the intestinal brush border, and then they can break down these dipeptides and tripeptides into individual amino acids. How many amino acids do we have? 20. 11 are essential, and 9 are non-essential, 9 and 11 are essential. And then here we have the enzymes that breaks down lipids: gastric lipase, pancreatic lipase, lingual lipase. And then we also have nucleases. That, I didn't mention it, but the pancreatic juice also has nucleases that breaks down nucleic acids and nitrogenous bases. And for the most part, they don't have a nutritional value for us. So that's all for this chapter. Do you have any questions?